In this video, we will review the anatomy of the eye and finish up the lecture material for the exam. So when you're looking at an overview of the eye, remember that your eyelids are referred to as palpebrae. And on this, you can see the outer canthus and the inner canthus. Those are just going to be the corners of the eye. And then we also talked about the lacrimal gland. That's your tear-producing gland. And as we mentioned before, it's producing tears, and they are going to move over to the surface of the eyeball. And then every time you blink, it's moving stuff across the eye, any kind of debris that's not supposed to be there. And then it's going to accumulate in this inner canthus. And all the liquid is going to drain down into your nose. And then a quick review of the layers or the coats of the eye. The sclera is going to be the outer layer. So in this view, it's the white that you're seeing here. That'll be the sclera. And then just to the inside of that is going to be the choroid coat, which is darkly pigmented. And then the retina is going to be on the inside. So this kind of orangey looking layer with all the blood vessels, that's going to be the retina. Now, just as a review, when you're looking at the sclera, which is the white of your eye, when you follow it to the front, it becomes more curved and transparent. That's going to be the cornea. And as we mentioned with the cornea, it's going to allow light to come in. So light's moving in. And it's also going to bend or refract that light. And that's really important because you want all of those light waves to converge at the back of the eye. And here's another view showing the sclera on the outside of the eye, and a really great view of the cornea at the front, and then you can see the iris in there as well. One of the functions of the iris that we mentioned, which is on the middle layer, it's on the anterior portion of the choroid coat. The iris, is what gives your eye its color. So whether your eyes are brown or blue or green, but there's actually muscles there, and this is showing you that those muscles are going to be contracting and relaxing to change the diameter of the pupil, which is the opening in the middle of the eye. Now the inner layer of the eye is going to be the retina, and in your notes it says that's the nervous coat. So we mentioned that it contains the photoceptors, which are going to be your rods and your cones. And they're just named based on their overall shape. So we're going to take a quick tour of the rods and the cones and then move on to new material. Now on the rods, we mentioned that there's approximately 100 million of these scattered all over the retina, and its job is to just give the outline of an image if you're walking into a dark room, and also for peripheral vision, so you can see things that are off to the side. We also mentioned dark adaptation, and just a reminder on the dark adaptation, this is when you're going from a light environment to a dark environment, and your eyes have to adjust for a minute before you can make out any images at all. So for dark adaptation, you've got to form rhodopsin. So we talked about that chemical equation, and the rhodopsin is what's going to activate your rods. So that lets you see better at night. And then if you go out into the bright light, that rhodopsin gets broken down so you don't have as many rods that are going to be active. So looking at the second kind of photoreceptors, which are cones, they have kind of a cone shape to them, and you don't have as many of these. And instead of being scattered all over the back of the retina, they are going to be concentrated on the fovea centralis, which is one specific spot at the back of the retina. So the function of these cones is going to be for color vision. So you only have 3 types, and I think I've mentioned this already. You have red, blue, and green. So it's every shade of color that you see is a different combination of those cones being activated. Now, the second function is provides a detailed image. So that's something like looking at the texture of velvet versus something really rough like tree bark, so that you can tell the differences between those. So this fovea centralis is going to be located on the back of the retina. In the macula lutea. So the macula lutea is a larger region, and it contains this little indentation called the fovea centralis. That again is where you're going to have the highest concentration of cones. And if you've ever heard of something called macular degeneration, this is where it occurs. When someone has macular degeneration, they're looking at something, let's say they're reading a book or looking at a computer screen, there's going to be a kind of a blank spot in the middle of the image. And it's because they're starting to lose those cones that are in the macula lutea. And that just gets progressively worse. Here's an image showing the back of the retina. You can see that it's highly vascular. And here is that macula lutea, and in the middle of it, that's where your fovea centralis is going to be. So high concentration of cones there. And that's the same thing that you're seeing on an actual shot of the eye over to the side. The optic disc is going to be located close to the fovea centralis, and at the optic disc there is no photoreceptors. So, because there's no photoreceptors there, if the image falls on the optic disc, you don't see anything. So that's also known as your blind spot. All right, so the reason there's no photoreceptors there is that that's where the optic nerve enters the back of the eye. Now looking at the inside of the eye, the lens is going to be located behind the pupil, and its job is to also refract or bend, it's the same thing, refract the light waves. So I know that you guys did the sheep eye dissection, and when you were feeling the lens, it felt really hard, almost like a marble. But in a fresh sample that hasn't been preserved, it's kind of squishy. It's still fairly tough, but it's squishy. It's going to be more elastic. And squishy isn't a scientific term, but you get the idea. Now, because it's squishy, it can change shape, and that's going to be the accommodation reflex. So, accommodation reflex, the lens changes thickness to view objects that are close or far. Okay, so if you're looking at something that's near, let's say you're looking at a computer screen or watching a video or reading something, your lens is actually going to become more rounded. And I'll show you an image of this in just a second. But if you're looking at something across the room, the lens is actually going to flatten out because it doesn't have to curve and bend those light waves as quickly. So Near or far? Near or far. That's the accommodation reflex. Now, on a cataract, if someone has a cataract, there's going to be protein crystals that form in the lens. So at some point, the lens actually becomes cloudy or opaque. And it interferes with vision. So a lot of people start to complain that what they're seeing looks kind of yellowish and kind of blurry, and then it just gets progressively worse. And at some point, if the cataract becomes extreme, they have to remove it. And what they'll do is replace it with a synthetic lens. And just a minute ago, I was talking about the lens being squishy. The lens becomes less elastic as you age. So there are people that have had to wear glasses for 15, 20 years, and then they have cataract surgery to replace that lens, and all of a sudden they don't even have to wear glasses anymore. So it actually helped with their vision as well as getting rid of the protein crystals. So here's a side view of the eye. Here's the lens that we were talking about. And I just want to mention that it's going to be attached to these little suspensory ligaments. And so they're going to stretch and then also reduce tension on the lens. So that's how it actually changes shape. Now, sitting in front of the lens and the pupil is going to be the anterior cavity. So all of this is going to be the anterior cavity. So we're going to write something down in a minute for that. And then behind the lens is going to be the posterior cavity. Now the anterior cavity, as I said in the image, it's located in front of the iris, and it contains aqueous humor. So when you're doing the sheep eye dissection, if it kind of squirted a little bit or had some very liquid fluid that seeped out when you cut into it, that was the aqueous humor. Now, that aqueous humor is actually produced by the ciliary body. Okay, and so that ciliary body is located on the back of the iris. Now, glaucoma is a disorder that can be genetic, and when someone has glaucoma, there's excess accumulation of aqueous humor. So that just means that the fluid isn't able to drain and there's still more and more being produced. And if it's not corrected, it back builds. There's more and more pressure that moves to the back of the eye, and at some point it actually starts to damage the photoreceptors and leads to blindness. Okay, so if not corrected, the pressure damages the photoreceptors and causes blindness. So that's why when you go to the eye doctor and they do that fabulous test where they put a little puff of air in your eye, what they're doing is they're measuring the internal pressure based on how much give the cornea has. So that's why they have you do that test. Okay, so here's an image showing how that whole process works. Here's the lens. Here's the ciliary body. That's where the aqueous humor is going to be produced, and it's showing that that fluid is being produced, and it starts to fill this entire cavity. And so you're constantly making this aqueous humor, and so you want the fluid to flow through this anterior cavity and then come out here through this scleral venous sinus. So it's going to move through there and migrate out and be reabsorbed. But if for some reason that vein is going to be blocked, then that's when you would have glaucoma, because more and more fluid is going to accumulate there. Now the posterior cavity is going to be located behind the lens, and it is a large cavity, and it contains a very thick, almost jelly-looking substance called the vitreous body. Sometimes it's called the vitreous humor. So that fluid is really important because it fills up all of that space in the posterior cavity, and it also helps to keep the retina in position. Now you will have to know the pathway of vision, and so this is just an overview. On the previous slide it actually shows you step by step the summary, so I'm just going to show you the same thing on this image. So here's a tree. Let's say you're out hiking in the forest and you see a tree. So all of these light waves are going to be moving towards the eye, and when they get to the cornea, which is that space in the front, again it's curved, and so it's going to refract the light. So then it's going to go through the pupil, through the lens. The lens is going to bend it even more. And then you want all of those light waves to converge on the fovea centralis. So that's the reason that the cornea is going to be curved and the lens is going to be curved, is you want to bend all of those light waves so that they fall right on the fovea centralis. And I think it's really fascinating that that image actually gets flipped and it's positioned upside down on the back of the retina. Same exact thing happens with the camera, same thing happens with the microscopes in lab. So let's talk about a few vision defects. First of all, normal vision is 20/20. Okay, and so we'll be looking at an eye chart and explaining what those numbers actually mean in a minute. If someone has myopia or nearsightedness, then they can't see very clearly when they're looking at distant objects, but they can look at something up close. So they can read just fine, but if they're driving without their glasses, they can't read the street signs. Usually they have to wear glasses for that. Hyperopia or farsightedness, they cannot see close objects clearly. They have hyperopia, they may have to wear reading glasses because of eye strain, but they can see distant objects very well. So here's the Snellen's eye chart, and just walking through this with you, here's normal vision, that 20/20. And if you look on all of these numbers, on every single one of them, the first number is a 20. Okay, so what that means is that when you're taking this test, you're going to stand 20 feet away. So that doesn't change. What does change is as you're going through here, they're looking to see what's the lowest line you can read accurately. Okay, so let's say that someone has 20/70 vision. That means that that person's standing 20 feet away, and that's the last line that they could read, but compared to someone with normal vision, they could back away to 70 feet and still read that line. Okay, same thing with, let's say, 20/30. If someone has 20/30 vision, they're standing at 20 feet, and that's the last line that they can read. But somebody with normal vision could back up to 30 feet away and still see that same line. All right, so what do you think is hyperopia? What do you think is going to be myopia? Okay, when you get down in this section, this is going to be hyperopia. So someone may have something like 2010 vision or 2015 vision. Up here, this is going to be myopia. So if someone's doing an eye test and they aren't wearing any kind of corrective lenses, no contacts, no glasses, and the only line they can read is this one, their vision is 2200. That's really, really bad. That means that they're standing 20 feet away and that's the only line they can see. But someone with normal vision could keep backing up to 200 feet away and they could still see that E. Now, if someone has an astigmatism, that's going to be an irregular curvature of the cornea, or sometimes the lens. Now, if someone has an astigmatism, they can still have 20/20 vision, but their vision can be blurred. A lot of people complain of seeing a starburst effect at night if they have an astigmatism. So what they have lenses that can accommodate for that so that it kind of smooths it out. There's even some contacts now that when they suction onto the surface of the eye, that it can actually reshape the cornea. So that's kind of neat. Now the last one we're going to mention is presbyopia, and that is going to be age-related farsightedness. So if you're looking at your parents or your grandparents, you may have noticed that they keep having to hold the newspaper or whatever it is further and further away from them just to be able to read it. That's presbyopia. So the reason that happens is that the lens becomes less squishy or less elastic. So they're losing that accommodation reflex. The whole near-far thing, they're losing that, and so they're more likely to have to wear reading glasses at that point.